about the first time that rather than someone forgetting and us not having a presentation, we actually have two people preparing a presentation.
I can leave my place if you want. I can.
It's fine. It's fine. You know what? I can be like a backup for another time, for next
Oh,
week or
okay.
when somebody wants to switch.
Next week we won't do Neuroccino because we're all doing our workshop in Bordeaux.
Because we're gonna be all reunited again.
I mean, us all. I thought you're joining as well.
Who?
You?
I'm joining, yeah.
Yeah, so
I'm
all of
coming.
us. Wow,
All right.
it is too early for you. Wow. Leah's joining us from the train again, it looks like.
Yeah, she's
Yeah, the train
traveling.
is late actually, so I'm on the phone. And yeah, I need to go in person in Parma.
Tough
But I'm
life.
listening.
Right. Okay. Misha's bouncing again. Welcome to Neuroccino. On the first day where we had two overlapping volunteers. But we're gonna stick with the schedule as we had it, as this was briefly sorted out. And we have Hervé. If you can share a link in the chat for us.
Yes, the link. Ah, the link.
The link,
Ah, okay.
famous link.
Let me
You can
find
all-
it very, very fast. Uh-huh.
And then we can all have a look at the
Yes.
paper.
Okay.
And then you know the drill. Take it away.
Yes. So let me share the link. So hopefully my computer will not play with me today because it's starting to behave erratically since last week, but I hope today it will be good. So let me partager, share my screen. Normally you should see my screen. And now, okay. And now, where is the chat? Hi here. Chat is here. It's a direct live. Yes. Okay. So you see.
We still don't have the link. Oh, there it is. Okay, cool.
Good.
We got it.
You got it. Okay. So today I'm gonna talk about this article, the emergence of genetic variants linked to brain and cognitive traits in human evolution. So just to come back to something, so because last year I think I presented an article which is really related to that, which is looking at human brain evolution. And one point I was making that is there is different ways of looking at the evolution of the human brain. The problem is the human thing and the evolution because we cannot go back in time. So there is different ways of doing it. So you can look at archaic brains, for example. You can derive, for example, the shape of the brain based on the endocast, which is a very indirect way of looking at the brain evolution. And if you are interested in cognitive evolution, it's difficult. You can look at primates also, so that are our cousins, and you can derive some information from it, but they evolve too, so it's not, again, direct way of looking at human brain evolution. And the third way is what they call the paleogenetic brain. So you look at the evolution of the genetics to derive information to the evolution of the brain. So this paper is looking at that. I think that, yeah. Normally what you do when you are interested in imaging, neuroscience, you look at brain phenotypes and you correlate them with genetics using what they called genome-wide association studies. So you take all the variants in the genome and you do correlation with brain phenotypes. So in the study I'm gonna present is what they do is that there is a way now to put an age on a SNP. So a SNP is a single nucleotide polymorphism. It's a variant. So you can put an age on each variance. And so this study is doing that. So doing that, you can go back to the age of the SNPs and go back to the brain phenotypes. So you bounce like Michel is bouncing from the gene to the brain phenotypes. So let's go back to the article and not this slide. Can I go to this slide? Okay, let's go back. Okay, so as I said, so it's what they're trying to do in this paper. So they collected, where is it? Where is the method? They collected all the SNPs available from 2,500 GWAS, so genome-wide association studies. So they took all these studies from a website where you can go, you can select the traits you are interested in, and they give you the SNPs, the variants, the genetic variants that are related to the traits you are interested in. So they got 33,000 SNPs, and from these 2,500 GWAS, and they derived the age of the SNPs. So let's go to the first figure. Okay. So you have this bunch of SNPs and age, so you can plot them. It's what they did. So you have the age here and the number of SNPs. As you can see, during the evolution of these SNPs, so I will not go back to how they derive the age of the SNPs because it's a little bit complicated, and I think we could discuss more about that. So you have two peaks in these SNPs. So you have a younger peak, which is around 50,000 years, and you have an old peak around 1 million years, okay? And so what they did also, they compared these SNP they got from the GWAS, so they called it phenotype associated SNPs. So just to come back to that, so the SNP the GWAS they selected are GWAS from, for example, eye shape, cancer, height, brain anatomy, and aspects related to cognition and behavior. Yeah, they wanted to stick with brain evolution. So they selected everything about brain. And they compare, they compare that with regular, let's say it like that. So regular SNPs. And so comparing that, you can compare and you can statistically see if there is a difference between these SNPs and the regular SNPs. So you can see this little star give you where you have younger, you have more occurrence of these phenotype associated SNPs compared to the new models. And you see that you have these, they are more representative of these phenotype associated SNPs in the younger stage and also in some periods. So that is the first results I got. So they compare also the minor allele frequency. So just to come back to that. So when you have a variance, you have a major allele and a minor allele. So the minor allele is, as it says, it's minor, it's less, it's rarer, and it's more rare. And what they found, but it's not something very extraordinary, is that the more you go, so when the variant occurs, at the beginning, the minor allele is very rare. So the frequency is very low for the minor allele. And the more you go, and the older is the SNP, the frequency of the minor allele will go up. So it's what they saw here. So you have more SNPs with a minor allele frequency, a lower minor frequency at the beginning, and after you have more SNPs with higher minor allele frequency, a MAF. Okay, they did that for two different databases. So okay, so let's just wrap up here. It's two peak, young peak, and old peak, and they old peak, and so they make a story about Homo sapiens is around this period, and so the younger peak will be more related to Homo sapiens, and the older peak with our related ancestors. So let's go to the fun part. So you have this bunch of SNPs with an age. So they selected all these GWAS. So all the GWAS are looking at a trait. So they correlate SNPs to a trait. And so you can group these GWAS to different categories. So they group them by chapter, domain, and traits. So you select the SNPs, you group them, and you look at the age of the SNPs. And you can, again, comparing if it is younger or older than the baseline. And you can look at that. So the different chapters are, for example, psychiatry, activity, environment, sociability, or neoplasm. So something I didn't know. So it's what they call neoplasm. The definition is a type of abnormal or excessive growth of tissue. So it can be cancer or it can be completely benign. So you have metabolite. And so that in this figure, they order this chapter by age. So for psychiatry, it's the variants are probabilistically speaking, are younger than the norm. So you have variants related to these traits, domain or chapter, that appears to be younger than the regular SNPs. And you can see that looking at traits, so you see depression, alcoholic drinks, again, depressed periods, or even schizophrenia. There are variants that are very young compared to the norm. So that here you have the older SNPs. And something it's fun is getting up in morning. So getting up in morning seems to be older. So don't worry, it's not our fault. It's our ancestors. We are not in charge of that. So something I was surprised is the cognitive, you have cognition here. And so it's not so much happening here. You have just the fluid intelligence score, which is below the threshold. But so on. And if you are interested in brain imaging, they put that in the neurology. Yeah, the neurology here. It's not a lot happening. Here, a little bit the same thing. Again, the psychiatry is in the young peak. And here, if you look at chapter level, you have mental and behavioral disorder in the young peak too. Okay. So another way of looking at them are these SNPs. So just before they group them, the SNPs per category. So what you can do is also to group them per gene. So the SNPs are distributed in the genome. They are in genes. So you can pick all the genes related to the SNPs. And what you can do is to give an age to each genes. So they did that. So here's the same thing as before. You have the young peak. You don't see so much going on after 50,000 years, but I'm not sure why. And so what they did, so I will focus on that. So they selected the 10 oldest 10% oldest genes and the 10 youngest genes. And they did what you call a gene-based enrichment. So you look at the genes, and so you see, so there are studies, databases that give you how much this gene is related to disorder or any kind of function. So they did that for different categories of function and disorder. So they look at schizophrenia, bipolar disorder, major depression, autism, Alzheimer's disease. They look also at brain volume, cortical area, cortical thickness, and also to intelligence and sociability. So what you see here again, so in yellow you have the oldest gene, and in red you have the young gene, and here you have the probability, the threshold for looking at how much they are represented, how much the odd of being young or old. So what you see is that for cortical area, you have a much younger gene as compared to regular age of genes. And for intelligence, you have younger genes. So it's a little bit complicated. So variants give you age, gene age, and so you have genes that are younger as compared to the norm. So looking at the 10 youngest genes, they look at the function. So they did functional annotations. They found that it's mostly represented in the function of the brain, so neurogenesis or the encephalon development. And so, and to continue, so when you have gene, you can look at gene expression. So they selected these 10% youngest genes and they look at the expression in the brain, in the human brain, and they found that it's mostly in, for example, in the pars opercularis, the pars triangularis. So they make a story about that on the language, because yeah, there are regions that are applicated in language. They discuss about it about the posterior cingulate cortex as it's a hub for the defaultment network. Just to continue, so they use neurosynth. So they selected this region. They look at neurosynth. What are the functions related to this region? So mostly, yeah, again, they found the language network, and everything seems to be not everything, but a lot of them are represented are in the language domain. And okay, so I will skip on that. So just to make short, these genes are more expressed during the prenatal period than in the postnatal period. Okay, so I hope you followed me. So I think that that is the last figure of the paper. So after they discuss about the results, so the main one is that they found variants, young variants that are related to psychiatric disorders, and they try to explain that. Why is it? Yeah, so human brain vulnerable to dysfunction and as psychiatric and neurodegenerative disorders. And they make a story that we evolve, with a kind of neurocognitive advancement, what they call cognitive advancement, but there is a side effect of that with a cost of this advancement with increased susceptibility for brain dysfunction disorders. So it's a balance between what we gain and what we lost. So in yellow, I put some of their topics in the discussion, which I found very cool. So I showed you in the first figure that some SNPs are related to alcohol, and I found that these variants are overrepresented in genomes of ancient farmers, but not in earlier hunter-gatherers. So agriculture seems to be linked to alcohol. Something else, yes. So genetic variants linked to psychiatric disorders seems to be also linked to ants' reproductive success. So because it's interesting that variants that are deleterious for us seems to progress, and they make that even if it is a bad thing, it's also maybe a good thing, saying that it's a factor for ants reproductive success. So there is a trait related to lifetime number of sexual partner, which seems to be the same variance as for psychiatric disorders. So let me make you the story behind it. I think, yeah. I was interested in this paper because they look at neuroimaging stuff. So they selected only studies. So at the beginning, I said that they selected 2,500 GWAS studies, but they selected only studies above 50,000 subjects. And that is a limitation in the number of neuroimaging studies you can get, because 50,000 subjects is a lot, and only one sample is available at this stage is a UK Biobank. So all the neuroimaging traits they got is from only one sample, which is the UK Biobank sample. So it's a limitation. I would have liked to have more. It's true that genetic studies need to have a big number, but I would have liked to have studies with a smaller number to include more neuroimaging stuff from other sample, like the ABCD stuff like that, with a more diversified representation of the population in the world. So. And on that, I think I'm done with it. I'm happy to answer your questions if I can do it, because it's there is a lot of genetic stuff which is I'm not familiar with too much. So, but
Amazing, very exciting science indeed. The chat has been on fire, mainly on technicalities of how do you do that. So I just give the floor to, I guess, the combination of Eduardo, Leah, and Michelle, who've been discussing the resources and how to do it.
That's it. We have to ask questions.
Well, you kicked it off with your question on the how, so ask our expert.
Hervé, I don't know if you can hear me. I have a question. Like, how do you define the age of the SNPs?
yeah,
Is
so
there...
that was the point of my. You were not there. So the last
sorry.
paper was discussing about that. So you compare. So you take the. So it's a comparative studies. So you have a genome from our ancestors. So from even from Neanderthals, Denisovans, and you look at. So you have an age of these samples, and you take the SNPs and how much the representation. So you have a percentage of minor. So I will make it simple, and even doing that, maybe I'm doing mistakes, but you have a percentage of minor and major allele. And so looking at different sample of ancestors, you can compare them with us, with our representation, the percentage, the representation of the SNPs in the current population, and you compare that with Neanderthal, Denisovans, and you can derive the age of the SNP. You can go to this.
It's like a colocalization. It's similar to a colocalization approach.
Maybe I'm not familiar with it, so. But yeah, I can just show you if you go there. They explain the methodology. Where is it? There is a website somewhere. Yeah, I put it up.
Okay, yeah, no worries. I go back to the other Neuroccino then.
But just to show you you can go to this website, which is human.genome.dating. And so you can select any kind of SNP. So you select on one, and it gives you the age. So it's looking like that is not really fancy, but yeah, you can have the age of each SNP in our genome.
Wow, there is a website for everything now. It's fantastic.
Yeah.
Mish also has a question now.
It's great, having. I had a great time. So I have two questions. The first one is that the first paper that shows that new SNPs are like more variables than the old ones and related to pathologies, or was there a paper before that was suggesting that?
Is there other paper? So that was, I think it was a strong hypothesis, but I'm not sure if they got other studies looking like that. It was a strong hypothesis that these variants were linked to younger variants, because it's part of our evolution. I'm not sure if there is paper like that dating each disorders like that. So no, I don't think so.
It's looking at the variability of neuroanatomy with Steph, we found and published that the regions that are the more variable in the brain in terms of their configuration are the more recent on the evolutionary
Yeah,
tree. And it's interesting to see that the same thing exists for genes. And
yeah.
maybe there is a framework to develop that related to pathology using neuroimaging. And the other one is like, so when we discuss in the chat, we saw that they're only going like up to 1 million years ago, which I guess you can have SNPs that are in a state that it can be explorable or analyzable orders than that.
Yeah.
But comparing species, do you think we could go further?
Yeah, so at the end, in the discussion, they say that so the comparative studies will be the way to go if you want to go deeper in this kind of analysis. Again, as I showed you at the beginning, it's a very indirect way of looking at brain evolution. So it's and there is at each step you can maybe make mistakes or approximation. And so, yes, they say that looking at other species will be also a way to corroborate or not these analyses.
Oh. There is no direct way of looking at evolution except traveling through time.
Yeah.
So
Or
I think it's good enough.
yeah. I mean now. Now I don't know. I didn't see a lot of studies going on that, but at one point they made a mini brain of Neanderthal.
Oh.
You don't remember these studies? I don't know if they end up still are they still doing it or not? But yeah, you could have a mini Neanderthal brain.
No, that's a brain.
So it's another
Exciting.
way to go also to clone.
I can't wait to have my own.
I was just going to say, don't tell him, he's going to add it to the collection. Eduardo has a question for you as well.
Yeah, I'd love to see a Neanderthal brain. But yeah, jokes aside, I was wondering like, if you know a little bit more on how they selected the traits they looked at, because yeah, cortical morphology, fluid intelligence, psychiatric disorders.
Yes.
It all looks quite human. So are we surprised that it's related to relatively young genes, or is this what they had available? They selected some, or how did they?
So there is a spreadsheet if you are interested. So it's a completely arbitrary selection. You have the traits, and after you do. So for example, let's go to results. So for cells. Cognition. So you have these domain, chapter, domain, and traits. So they were limited by the number of GWAS related to these traits. So you can see that some GWAS are on intelligence, verbal, numerical reasoning. So it's arbitrary, and also it's a melting pot. So each domain is a melting pot of a lot of things. So
Yeah.
I don't know what to
The
do.
mini mental.
Huh?
Yeah, there's the mini mental as well.
Yeah, so the MMS is here. Okay. So maybe not very specific. And as I said, for all the neuroimaging stuff, it's in neurological and it's not a lot of things. So metabolic, yeah, I would not go on, but yes.
Okay, yeah, interesting. Thanks.
Thank you very much for presenting this exciting paper and for all your questions. We are actually perfectly on time this week. So good to see you all. Have an exciting week in science and see you next Monday. No, 2 weeks.
2 weeks.
Bye.
Bye.